I keep hearing that CRISPR is going to revolutionize the future, the way that we cure diseases, the way that we eat, and even maybe generate new humans. But what does all of this really mean? My name's Haley, and today I'm here with a crash course on CRISPR. Before we can jump right into what CRISPR is, we first have to understand how DNA works, and especially how it pairs. There are 4 molecules that make up the basis of life: A, C, T, and G. Those are chemicals. They pair together to create bonds, and those bonds make up nucleotides, which make up your DNA. These nucleotides are ordered once they are put into the DNA structure. But if even one of these molecules is out of order, it could cause a variation, which could give a human a disease. A single nucleotide. That's crazy. Our DNA is like a set of baking instructions. If even one ingredient is messed up, it could cause a complete catastrophe. If even one of our nucleotides is messed up, it could cause diseases like sickle cell anemia. And that's why CRISPR comes in. CRISPR was actually discovered in E. coli. If we break apart the acronym, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. That sounds very complicated, but it actually tells us the 2 main parts of the system. Firstly, there's the repeats, which are short segments of DNA, between 20 to 40 letters in length. They're palindromes as well, which means that they read the same from left to right. That means that they have the same chemical letters on both sides of the repeat. Think of the word madam. You can read that from left to right or right to left, and you're still going to read madam. The repeats are all identical, but they're interspaced. So in the middle, we have spacer DNA. What's interesting about spacer DNA though, is that it's unique. This is because it matches up with viral or bacteria's DNA. Scientists also identified genes that were associated with CRISPR. Those are the Cas genes. They make Cas proteins that are able to unwind and cut DNA. So basically, in the E. coli that was studied, the system was like an immune system, but for bacteria. When a bacteriophage injects its DNA into a cell, if you didn't have an immune system, that cell would be hijacked. But what CRISPR does is create proteins, which is the Cas protein, and then turn the spacer DNA into RNA, which will fit into the Cas protein. This breaks apart the viral DNA and essentially stops the infection. Once the CRISPR system was identified in the E. coli, scientists were thinking about, well, what if we could hijack that system and then use it to activate the genes that we choose, or deactivate the genes that we choose? And so the hunt was on, and eventually, Jennifer Doudna and Emmanuelle Charpentier found a way to use this. They used the Cas9 protein. However, when they looked at it, it was in the S pyogenase. In the S pyogenase, there are not just 1, but 2 long strips of RNA. There's the tracer DNA that became CRISPR RNA, which is going to fit into the Cas9, and then there's the tracer RNA. The lab thought about using the Cas9 protein, but replacing the tracer DNA with their own sequence. They then merged the 2 RNAs together to get a tracer RNA CRISPR RNA chimera, which I'll refer to as the guide RNA. So, the 2 parts would be the guide RNA, which has the information on where we want to cut, and the Cas9, which actually does the cutting. If we know the DNA that we want, we can create a guide RNA that's going to correspond to that DNA. The DNA will feed into Cas9 to match with the RNA, and then the Cas9 will cut it. After it's cut, the DNA will be inactivated, or it'll try to repair itself. But if we want to insert a new gene, we have to add a 3rd part to our system, the host RNA, which is the RNA that we want to add to the gene sequence. So, as we break the DNA using Cas9, the host RNA is going to be added. Now that we know how CRISPR works, how would we apply it in the real world? Like I mentioned way at the beginning, sickle cell anemia is caused by 1 variation in the DNA. And since with CRISPR we can choose exactly where we want to cut, this disease could be completely eradicated. If you have any questions or you thought this was interesting, my links are down below, so feel free to reach out to me. I promise I don't bite.